we basically continue our conversation about biological molecules, and this one will focus on nucleic acids, such as DNA and RNA and their main components. Obviously, nucleic acids comprise an important part of human biochemistry. And first we're going to learn about the individual building blocks that make them up. The individual building blocks of any nucleic acid is a nucleotide. You can see the general structure of a nucleotide on the left here, okay? And it consists of three components: a nitrogen containing base or nitrogenous base, a sugar, and a phosphate group. Nucleotides are different because they contain different, there are multiple mistakes here, different bases. If we're talking about DNA, those bases are adenine, guanine, cytosine, and thymine. In RNA, adenine, guanine, and cytosine remain, but instead of thymine you have uracil. So now let's take a closer look at these bases. So bases can be of two types. They can be purines or pyrimidines. Okay? Out of 4 bases that can be found in any kind of amino acid, 2 types are purine bases, 2 types of pyrimidine bases. So you can see that pyrimidine contains one 6-member ring with 2 nitrogens in it. So the pyrimidine bases in the DNA are cytosine and thymine. As we mentioned, in the RNA, thymine is replaced by uracil. Now, what about purine bases? Purine bases in both DNA and RNA include adenine and guanine. Purine bases have 2 cycles fused together, the 5-member cycle and the 6-member cycle. So on top here, you can see 3 different pyrimidine bases. You can see that in each case, the pyrimidine nucleus, the 6-member ring, right, it has some kind of substituents, okay, it's cytosine. Uracil has different substituents, and thymine has different substituents. So just a little highlight, look at this. Uracil can be found only in RNA, while thymine can be found only in DNA. Purine bases are the same for RNA and DNA, and it's adenine and guanine. Again, you have this purine nucleus with the substituents to it. Does that make sense, everybody? The sugar, pentose, because it has 5 carbons, differs between DNA and RNA. The sugar in RNA is ribose. The sugar in DNA is deoxyribose. Let's take a closer look at what makes these 2 sugars different. First of all, I want to clarify something. It's sort of my small pet peeve. when I was studying this, when I was in the second year of college, it baffled me. Why are these carbons numbered 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime? Do you see that numbering? Well, the explanation is actually very simple. Let me roll back a little bit. Do you see this nucleotide? See that? There are carbons in the sugar, and there are carbons in a nitrogenous base. Do you follow my train of thought? So these are carbons in the sugar, right? While these are carbons in a nitrogenous base. So the carbons and nitrogens in the nitrogenous base have numbers 1, 2, 3, 4, 5, and 6. The carbons in the sugar have numbers 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So we can distinguish them, the carbons, I mean. Is that clear? Let's go back to this one. So this is the comparison between the RNA and DNA. So in RNA, you have this hydroxyl group in 2 prime position. In DNA, you don't. That's it. That's the whole difference between ribose and deoxyribose. Am I making sense? Why is this important? This one single OH group makes RNA molecule very chemically unstable, and we will sort of talk about why this is important when we get to protein synthesis. Okay? Good, everyone? So when you combine a sugar and a base, this combination is formed a nucleoside. Okay? So nucleoside, nitrogen atom in a base, connects to carbon number 1, 1 prime carbon, via N glycosidic bond. Okay? So, how do you change the name? So we have adenine. Okay? Let me show you. So we have a base called adenine. Can you see that? We have a sugar, ribose. When you combine adenine and ribose, you change adenine to adenosine. Okay? I'm going to ask you a question then, using the analogy principle. So, if you have the base guanine, what's going to be the name of the nucleoside that this base can form? If adenine becomes adenosine, guanine becomes, okay, you change INE to ozine. So, guanosine, yes. Okay, makes sense? Now, for pyrimidines, let's say you have thiamine. Thymidine. Okay? Good? Uracil. Uridine. And finally, cytosine, cytidine. Okay? So far, does that make sense? So, terminologically, if we say the word adenine, we refer to a base. When we say a word adenosine, we refer to a nucleoside. When you combine a nucleoside with a phosphate group, you produce a nucleotide. In our particular case, the phosphate group binds to the 5' carbon of either ribose or deoxyribose. Okay? And it's really important. It's only 5' carbon to which phosphate group connects that will give the molecule the designation nucleotide. Okay? DNA and RNA contain only 5' monophosphate nucleotides, and I intend to spend a lot of time on this image. Okay? Good? Hear me out. What is this? What is this? A base, right? Adenine, a base. Okay. Good, clear? What is this? A base plus a sugar. Right? We call it a nucleoside, and the term will be adenosine. Am I making sense? When you add phosphate to the 5' carbon, you get adenosine 5' monophosphate. Good? No? Not clear? Yes, absolutely. Now, Ariel, sort of repeat after me. Okay? No, seriously, because it really helps learning. What is this? The base. And the base here is adenine. Good? What is this? No, all together. The sugar plus base is what? A nucleoside. Okay? So, let me do it slightly differently. I'm going to pick a different color so it will be probably clearer. This is a nucleoside. Agreed? Adenosine. Okay? What is this? It's phosphate. So we talked about esters before that are formed between an alcohol and an acid. Okay? So phosphate is pretty much comes from phosphoric acid. And I'm going to make acid capital. Okay? Now if we scroll back to this, This is the hydroxyl group. Hydroxyl group is characteristic for an alcohol. Does that make sense? So when you combine acid, sorry about that. Oh God. Okay, acid, phosphate, right? Do you follow? Plus alcohol, OH group. What you're going to get is an ester. Okay? So this is the ester bond. Better? Here's another question that I'm going to ask you. It's a simple question. Are there more hydroxyl groups in the sugar? Yes. But it's going to be a nucleotide only if phosphate is sitting at the 5' carbon. Only in this case. Making sense? Now, you can see that the only difference in naming, if we have a sugar that is ribose, it's going to be adenosine 5 monophosphate. Mono means just one phosphate. If we have a sugar that doesn't have hydroxyl group, in other words, if we have deoxyribose, it's going to be deoxyadenosine, deoxyguanosine, deoxycytidine. Okay? Good? Naming conventions? Clear? Okay. So this summarizes the components. I'm going to highlight the differences between the two because other than that, they are very similar. Sugars are different. RNA has uracil instead of thymine. Makes sense? And obviously the difference in nucleotides is going to be sugar. In case of base, it's going to be uracil in RNA and thymine in DNA. But other than that, structurally, they are very, very similar. Questions so far? Okay, we talked about it. So when you name a nucleotide, if it's a DNA nucleotide, you add deoxy and always 5' monophosphate. Okay? Good? Now, I want to scroll back. And I want you to take a look at, let's say, well, all of them, really. If you take two nucleotides, let's say you take this and this, cytidine 5' monophosphate and uridine 5' monophosphate. Which part of each nucleotide, which part is different between the two? The base. The base. This is why when we explain the sequence of nucleotides in a molecule of RNA or DNA, instead of these long and cumbersome names, we can just use letters A, G, T, C, or U. Is that clear to everybody? Okay. So this two tables, contain all the names for the bases, right? Names for the nucleosides, which you can see in DNA, are going to contain the prefix deoxy, and names for nucleotides. Okay? Now, I'm going to ask Ariel again. I'm going to repeat my question. So in all these nucleotides, this phosphate, to which carbon is it connected? 5'. Are there other hydroxyl groups except for 5'? If we connect phosphate to any other group, will it be a nucleotide? No. This is why we can omit 5' and just go deoxyadenosine monophosphate. We don't have to say 5' monophosphate. It is implied. Okay? So, let's do this. RNA, DNA, or both. Guanine. Adenine. Thymine. Uracil. Need new pen. Okay. Let's talk about primary structure. Okay. It's basically the sequence of amino acids, but we need to look closer at the structure. Right. In the nucleic acids, 3' hydroxyl of one sugar bonds to the phosphate on the 5' carbon in the sugar of the next nucleotide. I'm going to go between the slides to illustrate my point. Okay? Take a look at the right image. I'm going to make a series of statements and you're going to tell me if you agree or disagree with me, okay? So this phosphate is connected to the 5' carbon. Everybody agrees with that? This sugar, yes, this sugar has hydroxyl group at the 3' carbon. So this oxygen attacks 3' carbon, okay, releasing water and forming the second bond. This phosphate group is it connected to 5' carbon of the sugar we're going to give it number 1? Is it connected to 5' of the sugar 1? Is it connected to the 3' of sugar 2? So it is a universal rule that a phosphate connects 3' carbon of one sugar and 5' carbon of another. Okay? Is it clear? This is called, interestingly enough, correctly called, phosphodiester linkage. To be completely clear, in some sources, it will state that it is phosphodiester bond. It's incorrect. There are 2 bonds there. It's just a conventional way to call this phosphodiester linkage. Am I making sense? Okay. All right. So this sequence of nucleotides is called the primary structure. Okay? And I want you to acknowledge something else here. If you will be building this sort of line of nucleotides and adding more and more, you're going to have one end of that chain having a free phosphate. Clear? You have another end of the chain having a free hydroxyl. Can you see that? So this end of the chain is called 3' end, and this end of the chain calls 5' end is called 5' end. Make sense? On the 5' end, you're always going to have phosphate, and on the 3', you're always going to have an OH group. Okay? Conventionally, look at this. We don't have to picture all of it. Okay, because phosphates and sugars are the same. All we need to designate is the bases, right? And we need to put them in the correct order. So, scientific convention, hear me out. If you have a sequence A, C, G, U, A, C, G, and U, the 5' end is on the left, the 3' end is on the right. So if I say I send an email to my colleague, and in that email I put a sequence, something like GCG, UA, CUG, CA, if we kind of, have the same background, the person who reads this sequence, they know it's a 5' and it's a 3'. I technically don't have to specify. So, which base in this example is on the 5' phosphate end? C. When you said A, I thought sorry, A is in the middle. Yes, but the answer is A. Yes, cytosine. No, it's fine. It's fine. Okay? Now, the major difference between DNA and RNA, another major difference, is that RNA has just one strand, one chain of nucleotides. DNA has two. So in the 1940s, British scientist, I think he was Michael, last name Chargaff, he was looking at the chemical composition of nucleic acids, okay? And he discovered that if we take a large enough molecule of DNA, the percentage of adenine that he could isolate was equivalent to the percentage of thymine. And the percentage of guanine was equal to the percentage of cytosine. That makes sense to you? It was uniform rule. Let's say you take human DNA, and I'm spitballing, okay? You take human DNA and you discover that it contains 23% of adenine and 23% of thymine, and 27% of guanine, 27% of cytosine. Do you follow my train of thought? Okay, I'm going to write it down. Okay? And same here, for example, 27% of G and 27% of C. Right? Reasonable? Okay, it's called Chargaff rule. So in 1953, I'm going to correct some. It's actually a very interesting story of how it happened. So James Watson, an American scientist, Francis Crick, the British scientist, and Maurice Wilkins, also a British scientist, they were working, I think, at Oxford, either Oxford or Cambridge. They had another scientist called Rosalind Franklin, female scientist, and she analyzed DNA molecules using X-ray crystallography, X-ray diffraction. And by the results, she said that DNA molecule must be a double helix. And Watson, Crick, and Wilkins quickly realized, let me just scroll up here, that it will explain the equal ratios of A and T versus G and C if we presume that every A pairs with T and every G pairs with C. Okay? So the idea was that we have two strands. I'm going to make a very, very crude depiction of this. Right? And if one strand contains C, then opposite to it in another strand, there will be G. If one strand contains A, in another strand there will be T. Is it clear? It's a double helix, okay? And these lines effectively represent sugar phosphate backbones. I'm going to show you an illustration in a minute. So far, making sense? Now, they try to, there is a photograph, I don't know if they have it here, a famous photograph of Watson and Crick sitting with like a human-sized model of a double strand. They didn't do it, well, they probably did it because it looked cool, it did, but also they were physically trying to fit the bases to each other. They were trying to see how they can be combined. Am I making sense? And they figured that the only way this model would work, if one strand and another strand are anti-parallel. Does that make sense to you all? Well, we'll get there. So basically, if we will try to depict it in like these strands, so top strand will go from 5' to 3' from left to right, and another one will go 5' to 3' from right to left. Okay? Now, they published this paper in 1953. It was one of the most cited papers in the history of science. It's 1.5 pages. For that, it was published in Nature. They received a few years later, Wilkins, well, not a few years, some years later, Watson, Crick, and Wilkins received Nobel Prize. Franklin, so that paper, which used Rosalind Franklin's results, did not have Rosalind Franklin as one of the authors. Guess why? Yes, because she was a woman, and she absolutely deserved a Nobel Prize. But unfortunately, she died of cancer very early because she worked with X-ray diffraction and did not use proper radiation protection, developed leukemia, and died of leukemia. Okay? One of the really sad stories in the history of science. Okay? So, as we mentioned, this base pairing is called complementary base pairing. So on the left, you can see the pair between adenine and thymine. Okay? Does that make sense? So what happens here? Why do they pair? Because there is slightly positive hydrogen and slightly negative oxygen. Remember, oxygen is very electronegative, so it pulls the density, electron density to itself. Here, slightly negative nitrogen, slightly positive hydrogen. So we have hydrogen bonds. Am I making sense? Good? Now, when you look at the pair between guanine and cytosine, there are 3, not 2 hydrogen bonds. Which pair do you think is stronger? Yeah, the triple bond. And actually, you can predict, to some extent, the function of various DNA fragments. If they have a lot of GC pairs, they're going to be ridiculously hard to separate those strands. If they have a lot of AT pairs, they're going to be much easier to separate, and maybe that's what they need it for. So basically, we have 2 strands of nucleotides that are held together by hydrogen bonds, 2 bonds between A and T, and 3 bonds between G and C. So look at this illustration here. This band, the purple band, represents sugar phosphate backbone, in which sugar molecule, phosphate residue, sugar molecule, phosphate residue, they go one after another. Inside of that double helix are paired bases, GC or AT. Clear? So let's see how well you remember the base pairing. What should I put across from A? Just keep going. C, G, yes. Common question, are there other bases? Yeah. It's like minute fractions of percentage. There are some other bases included. Now, another question, can there be mispairing? Yes, and it's not good. It distorts the DNA structure, may lead to mutations, or it can be fixed by your cellular enzymes, because DNA must have pairing. So what is the function of DNA, folks? No, that's too broad. What is DNA? Like what is its function? It stores the genetic information, yes. Why do we have 2 strands? What is the biological importance of having 2 strands? Okay, what you just did, you had the entire information in 1 strand. Do you agree with me? And based on that information, you created a 2nd strand because of the base pairing rules, right? Do you follow? So why do you need 2 strands? Huh? Okay, why do you need a copy? Why do you need to recreate? Common sense. It's backup. If 1 strand gets damaged, information in the 2nd strand will allow you to recreate to fix the damage. Does that make sense? Good? Okay. Now, we mentioned that, DNA is storage of the genetic information. Why are your kids, why do they look similar? Like, why do they look not exactly like you, but they look kind of like you? Why do your kids look like you? Yeah. So you have to pass DNA to your offspring, whether you are a multicellular organism like humans or you're single cell, right? You have to pass that genetic information. But if you're a cell and you have DNA and you want to pass it down, you need to make a copy, because a cell will produce two cells, right? So that copying of DNA is called replication. Before the cells divide, DNA is copied. Okay? So how do you copy it? Strands of the parental DNA are separated. Can you see that separation? Okay? So the enzyme that separates them is called helicase. Okay? Breaks down the hydrogen bonds, and strands are separated. And then each parental strand, do you see the purple band? Okay? So these are parental strands. Okay? And the blue ones are newly synthesized. Does that make sense to you? You can see that there are nucleotides added to the growing chain. We're not going to go into exact details. But effectively, look at each daughter DNA. The daughter DNA, does it contain one strand from a parent? Does it contain newly synthesized strand as well? So that way of copying, when you split them and each new double-stranded DNA molecule contains one parental and one daughter, it's called semi-conservative replication. Okay, sounds fair? So the enzyme that adds those nucleotides is called DNA polymerase. Sorry. So at the end of the replication, okay, you're going to have two new strands, and they ideally are the same, identical, that contain the same information as the parental DNA. And the reason why we can copy with such great precision is complementary base pairing. What you just did before, remember in that learning check? Okay? Now, okay, we're going to finish. I'm going to talk about something. I'm going to record it specifically. I came up with this analogy. Trust me, it's going to be very useful in any biology class that you're going to take. I know. Are you going to take biology classes in the future? I talk like A and P and stuff like that. Yeah. Okay. So hear me out. I'm going to write something here, because this piece of knowledge will be very useful when we meet next time and talk about it. So you have DNA. DNA of human DNA consists of roughly 25,000 genes. So imagine that DNA is your family collection of recipes, like a massive recipe book. There are dishes that your great-grandparents cooking and stuff, okay? It's really huge. Do you follow my train of thought? So, you have 25,000 recipes. Your friends are coming over. They called you. They say that we're coming over for dinner, okay? One of your friends is lactose intolerant. One of them has celiac disease. And there is a Jewish kid who obviously doesn't eat and there's a vegetarian friend. So you need to pick dishes that will satisfy all those different tastes. Clear? Are you going to cook all 25,000 recipes? Probably not. So you're going to pick something, few recipes, that will work for that company. Okay? But here's the problem. This is your family recipe book. First of all, it's huge. It's enormous. Second, it's your family heritage. If you bring it to the kitchen, you can spill something on it or accidentally, I don't know, set the page on fire or something like that. You don't want to do that, right? And it's huge and cumbersome and inconvenient. So in the real life situation, when you have big-ass recipe book and you need like, I don't know, 6 recipes to cook, what would you do? How do you get those recipes to the kitchen? Okay, new generation, yes. Take a picture with your phone, right? Or, well, nobody writes it on a piece of paper. So take a picture on your phone. Am I making sense? So that picture is RNA. Remember, it's not a dish. You see what I'm saying? Like, people are not going to eat those pictures. We bring it to the kitchen and you use these recipes, the instructions, to make an actual meal. Okay? Is it clear? So, why this is a perfect, like seriously, I know I'm bragging about this, but perfect analogy. If you have a mutation in DNA, you have a change in the recipe, your dish may come out different. Is it clear? Remember what I said about which molecule is more stable. I said it about 30 minutes ago, DNA or RNA. DNA. It is the heritage. It has to be stable. RNA, we had a joke in the lab when we worked with RNA. If you look at RNA the wrong way, it disassembles. When you work with RNA, you work on ice as fast as you can. It degrades immediately. Okay? But think about this. Wouldn't it be nice if you take a picture of the recipe, cook it, next day it's gone automatically? You see what I'm saying? You don't need those pictures. They shouldn't be in an archive. Clear? And most of the cellular functions are carried out by proteins. Just like your friends aren't going to eat pictures on the phone or pages from the recipe book. They're going to eat a dish. So in order to do something, cell has to go all the way down to the protein. I'm dead serious. If you remember that analogy and you put it to use, cell bio, microbiology, to some extent NP1, NP2, super helpful. Okay? Cool? All right, folks. I'll see you next Monday.